1, 2, 3.
Okay. Coming to the next talk, and I'd like to introduce Alexandra BattagliaMayer from Rome. She got her degree in mathematics from that university, and then she got a PhD working already on the motor system in monkeys, and then she did a postdoc time in the lab where one has to go if one wants to work on reaching and arm movements, which was the Minnesota to Georgopoulos lab. She returned to Rome to start her very, very successful work. Many important findings. We already saw one presented by Roberto yesterday, and I think your work on the error correction is absolutely fantastic. And now we will see what happens if two monkeys interact.
Okay, thank you very much for this nice introduction. And also, of course, thank you very much for this privilege to speak here in front of this outstanding and warm audience. Thank you for this invitation to organizer. Okay, so speaking of mystery of the brain, I think that one question could be what makes human a different kind of animal? So I will start to approach this question from an anthropological perspective. And from this point of view, there is no doubt that having a bigger or smarter brain can explain our success in our evolution, but rather this success seems to reside in the ability to perform a collective behavior, because we do know that we can achieve goals, as the images here can recall, that goals that single individuals could never achieve alone. Of course, this collective behavior is not a prerogative of our species, but there are many examples. I mean, we have seen, for example, in the test that Nikos shows in his slides, in which a very simple species like animals like insects or even bacteria are able to show this collective behavior. However, in our case, in humans or at least in primates, what can be indicative of a different kind of collective behavior is our ability to predict or to adapt our action to those of the others during our interaction, at least motor interaction. As you will see in my talk, I will focus on particular type of interaction that are confined to inter-individual motor coordination. Despite the importance of this motor interaction and the fact that on the daily behavior we do move not only alone but with other people, in the last 60 years motor function has been studied in single brain action, in a solipsistic fashion. Since at the very beginning, from the works of Evert, at least focusing on monkey brain, on primate studies. Since the first works from Evert, and that was studying movement, start to study movement in one dimension at the single joint level. Then this study has extended from, has been extended in movement in 2 dimensions, then in 3 dimensions by the works from Georgopoulos, by Roberto Caminiti, and reaching in time studying even more complex behavior like those mimicking handwriting, or even more complex tasks like solving maze, copy task, until the end to study which is the the mechanisms that unify action perception and action execution with the discovery of the mirror neuron system. So at the end of this timeline, I thought it was time to understand, to extend this dimension, studying the motor function in the space in which we do have to interact with other individuals. Of course, when we speak about motor interaction, we use a term that is joint action. I will not go in details for time reason, of course. In literature, there are different examples, particularly in studying human behavior. Speaking of joint action, these are all the main papers that I have selected for this purpose. But despite the fact there are different examples of joint action, what makes an action which feature distinguish joint action from events involving multiple agents who are merely acting in parallel. So I will go straight to the definition, for at least for the purpose of this talk. We will define joint action as any form of motor interaction whereby two or more individuals sharing an intention coordinate their action in space and time to achieve a common goal. So we have different keywords here: motor, sharing an intention, coordination, space and time, and common goal. So all elements that are important for the purpose of this talk. So to study this aspect, we have used a paradigm working with monkey. I will describe later the task. And let me, of course, underline the fact we are speaking of a particular kind of joint action that is not, of course, working with primate, a non-verbal joint action. That is the fact that language, in this case, does not provide any cue to help the individuals to perform the task. That could be an issue if we want to coordinate our action when we want to bring a table from one part of the room to the other. And speaking of definition, it's also important to, I will sometimes speak of cooperation, even if cooperation, at least from an economical perspective, a cooperator is someone who pay a cost C for another individual to receive a benefit B, and while a defector has no cost and does not deal out benefits. While coordination is different, can be defined as an orderly alignment or adjustment of partners' action to achieve jointly some specific goals. And when we speak about coordination, it's important to distinguish different types of coordination. There are sometimes emergent coordination in which these emerge spontaneously between individuals that actually have no specific plan to perform action together, as the picture here, the people at the football game or when you walk on the street. And in this case, different sources can lead to the emergence to the emergence of similar behavior, but without having any specific plan. While a planned coordination, that is actually the one we are interested in, requires specific plan that specify and influence the joint action outcome. For this purpose here, we have this design a task in which the two monkeys here sitting next to each other are performing a task manipulating a joystick that guide cursor on a screen that are in front of the two monkeys. And so they have to solve a task in two different conditions, a solo condition and a joint action condition. So the idea here is to compare different action in two different contexts that actually are identical action from a motor perspective, because they have to apply a force on an isometric joystick that is represented here, but the same identical action has to perform individually or in coordination with the other animal. During the performance of the task, we have recorded simultaneously from different areas. In particular, here is I have represented premotor cortex. Should be okay. In the next, when I will describe the neural data, I will show you also the recording sign from parietal cortex. The idea is that we are recording from these two brains while they're performing the task from homologous areas of their brain. The main question among the different that we can ask in this type of task that I have selected for this talk is, of course, the one from that we can understand from a neurophysiological perspective is, does the neural activity differ when an identical action is performed individually or together with a partner? And more importantly, is the neural representation of joint action simply the sum of the neural representation of individual actions? So this brings us to an observation that has been made by the American psychologist Woodward in the 1939, that at the time was approaching the problem of studying at the behavioral level, of course, the joint action. And that for me is interesting because it could be useful to understand the data that I will present later. The concept of constraint has a great importance, not always recognized at the time, in the analysis of social behavior. The constraining factors need to be considered as well as the active individual. So the question that arises from this observation, from my point of view, is, are these constraints encoded at cortical level? Let me describe the task on which I will focus on. The monkey has to perform the so-called center-out task, applying a force on the isometric joystick, and they have to bring their own cursor. Each monkey is assigned one color. The green monkey has the green cursor, the blue monkey has the blue cursor, and once the peripheral target is light in one of the 8 peripheral position, the green monkey has to move the cursor if the circle is green. The blue monkey has to move its own cursor alone, individually, nothing new from the previous literature, towards the peripheral target. So these are the example: green monkey if the target is green, blue monkey if the target is blue. But the new part of the task is that when the target is white and a yellow circle appears that represents the common object that the two monkeys has to bring towards the peripheral target, coordinating the forces represented by the movement of the two cursors in such a way, coordinating in space and time in such a way to bring the yellow object, the common object, towards the peripheral target. So when the target is Y, they have to bring this cursor together. So, of course, the monkey do not work for free, and they will be rewarded if they do this task successfully. The reward that is not modulated is identical for the solo condition and for the joint action condition. And they are rewarded simultaneously only if they coordinate together the task to bring the object on the periphery. Here are examples of real trials. A green monkey is moving, just one trial. And here is the blue monkey is moving towards the peripheral target. I would like that you appreciating the velocity profile that could be indicative of how this individual action could be different between the monkeys, despite the fact that they are very well trained. In the case of the blue monkey, you will see the velocity profile peaking at this time, while for the green monkey, the movement is peaking at much very late, I mean the velocity profile. While for the case of the joint action task, you will see how they are coordinating in space, of course, their movement, and they have to align in time the velocity, the speed in with which they apply the force to bring the cursor to the periphery. Of course, we, being behavioral neurophysiology, we are obsessed, as you probably know, to control behavior, and we were controlling also the eye movement during the task. And this is the eye movement in the A direction during the joint action task. So a preliminary question is at the behavioral level. So because we are working with monkeys, this task is pretty null, so we didn't know at the beginning if these tasks, they were succeeding in this task just as an emergent coordination because the monkey are overtrained, so there is any evidence, objective evidence that the subject are actively cooperating in our task, and which kind of a coordination, planned or emergent coordination, intentional or non-intentional. So there is evidence that the different parameters are modulated during these tasks, and there is evidence that during the planning phase, for example, during the reaction times, they change their strategies to solve the task. So I quote Mickey Goldberg's sentence, since in behavioral neurophysiology, one monkey is an anecdote, in two monkeys is a science, if I'm not wrong with this, we test is 6 monkeys, 3 different couples of monkeys to test the behavioral data. And these are briefly the we tested different parameters. I have selected some. First of all, the reaction times. These are the different monkeys, 3 different couples, reaction times. Blue is the solo condition, red is joint action condition. And there is clearly a repeated pattern in which the monkey tend to generally decrease their reaction time. They are significantly shortened, with an exception of only one monkey that actually make the movement slower, but this is okay, I will tell you later about that, but this is the only case in which the monkey in the individual condition is is much faster than the other. So this seems to us the only way to make the reaction times closer to the one of the other monkey, and I will give you evidence of a learning process for this monkey to try to be more similar to the other monkeys. So just this to say that there is evidence of a process that is implemented during the planning phase. But also during the execution, the movements are modulated because the monkey tend to be slower. The dynamic force time, that is the time in which they apply the force, are much longer during the joint actions task, and this corresponds to a decrease and to a slower down of the force application during the joint action task with respect to the case. This is an example of a speed profile with respect to the solo condition. This is on the temporal domain. At the spatial level, the trajectory are in the joint action task are much more similar and straight with respect to the solo condition, in which they are more spread, and so there are more degrees of freedom. And at the same time, if we measure the intercursor's distance in the joint action task, comparing with trial in a similar direction, we see that the intercursor distance is much closer, is much shorter in the joint action task. So the monkey really tried to be closer during the joint action trial with respect to the solo condition. So an evidence of an active inter-individual motor coordination. And in this case, instead, this could be an evidence of the monkey being more predictable when they solve this task together. So speaking of the exception of the monkey that were the only one that was increasing, this is an example in which there is evidence of a learning process. These are the different sections in which the monkey has been tested together from the beginning to the end. These are an example of reaction times for the green monkey in the solo condition. The other is the monkey in the joint action condition, monkey K, monkey, I don't have glasses, K in this case, monkey S in this other case. So this monkey that was the faster, clearing time tend to be more closer in reaction time to the other monkey. And this seems to fit quite well. This is not only, this is just an example, we have other evidence with the decrease of the error rate in solving the task in the joint action condition compared to the solo condition in which the error rate is pretty stable. So there is this evidence of a learning process aimed at minimizing the difference between the two behaviors that results in maximizing the performance during the joint action task. So the conclusion for this first part related to the behavioral results could be that to answer the question if there is evidence of motor coordination, both action kinematics and kinexis of each monkeys seem to be reciprocally shaped by joint action. And how this is done, so there are cooperation invariances, that is repeated patterns of change of behaviors across the different couples, so shortening of reaction time, elongation of action execution times, and in general evidence of an increase of spatial coordination. So all these point to strategies that tend to increase the predictability. So I act in a way that are more predictable to facilitate the job for the other person, the other individual, to solve the task with me. And then the final question is an emergent or a planned joint action. The joint action performed in this task is, we saw, is modulating the reaction time, so is planned in advance, and it is not only emerging from the constraint imposed by the task. Therefore, now it's time to go to the neural data. We have recorded I realized that I made it biscay because cutting and paste a picture is not appearing. That we have recorded the neural activity simultaneously from premotor cortex and from area PFG of the two monkeys. So the simultaneous recording from in session from the frontal cortex, and in other sections simultaneously from the two parietal cortex. In particular, we were recording from area PFG, inferior parietal lobule, area 7, for those who are more familiar with Brodmann classification. And I will present the data from frontal and parietal cortex simultaneously to compare the two patterns of activation that we found. First of all, what we have found is what we could simply expect, that our cells here are example of cells in premotor and parietal cortex. The raster, of course, are representing action potential. The two groups represent three different directions of movement. And in the three different conditions, the solo condition, the joint action condition, and also the condition during which the monkey observed the other monkeys moving the cursor. Here is the behavior, the velocity profile, to indicate that during the observation there is no movement and no application of force that is present in a similar fashion during the solo action and the joint action behavior. So the two actions are very similar during the force application, and the two activities are compared to, if you are not convinced that they are similar, of course, these are statistically assessed, these similarities, and so we call these action canonical action cells, so simply related to the action per se, regardless of the context in which they were performing the task. And but fortunately for our experiment, we also found a nice modulation that was observed when the monkey were performing the same action, but only the cells were strongly modulated both in premotor or parietal cortex when the monkey were performing the joint action task. This can be depicted even in this task, in this plot, in the form of polar plot in the 8th direction. The red curves indicate the strong activation during the joint action task. And a similar pattern is for this other example in parietal cortex, also in the form of spike density function. And this is, of course, an example of two different cells. What we can see at population level is for each line here we have one particular cell, in the case of for a premotor cortex. Here are the canonical action cells, the joint action cells defined on the basis of their significant difference when we do compare the two different conditions. And I would like that you pay attention to the switch from these slides to the other, because this could make the difference between the main difference that we found from premotor and parietal cortex. The two activities apparently are very similar in the two areas, but when we switch to the same graphs, to the same plot in parietal cortex, you could appreciate, but it will help you with another plot, the fact that the activation of parietal cortex is much later with respect, I plot depicting here the peak of activation. This was the one relative to the previous graph on premotor cortex. And so what we observe is that the joint action cell, they generally tend to predict and to be active much earlier with respect to the parietal cells. We have performed an ROC analysis to see how this joint action cell discriminate the two different actions, and particularly when this activity was particularly the activity was discriminated, in which phase of the task. These are different, the ROC values, actually the area under the ROC curve, for different cells in each direction, in premotor and parietal cortex. Here is the mean of the ROC values in premotor cortex for the JA cell, for the joint action cells with compared to the canonical action cell. And we see that the discrimination power, is significantly different from the solo action cell during the reaction time. The 0 here is the appearance of the target. This is the start of force application. So during this phase, the premotor cell start to discriminate the difference between the two tasks. While for the parietal cortex, for the parietal cell, the discrimination is only during the execution phase and not during the planning phase. At this point, the question could be, which is the functional difference between the joint action cell and the canonical action cell? To answer this question, we have performed a multilinear regression with two different models. We place tip to tail the activity in the 8th direction. The blue curves indicate the spike density function of each cell. Here is there is the example of 3 different neurons, and we model this activity with 2 different models. One relative to the action of the individual from which we have recorded the activity, and that is indicated in green, called on action, and also with the parameter that is with the behavior, I will tell you which parameters, of the other monkeys. In particular, we place in this model the position, the direction, the velocity of the cursor of the 2 monkeys, on and other. And this, of course, is example of a very good fit and the good performance for this regression analysis. And what we have noticed from this test is that in premotor cortex, here we plot the R² in the form of cumulative distribution, and the shift of these curves means that there is a better performance for the joint action cells with respect to the green curves that indicate the solo action or canonical action cell. And these graphs indicate for both premotor and parietal cortex, in particular for premotor cortex, the joint action cells provide a more accurate representation of the on and the other action respect to the canonical action cells, while in posterior parietal cortex, this better representation is confined to the on action, while for the other action, there is no significant difference in this neurorepresentation. And another aspect that is important is on the temporal domain, as I say, and comparing the time lags that we have computed from the regression analysis and comparing the premotor and parietal cortex, we see that we have a higher number of cells with negative time lags, both for when modeling the on action and the other action. So in premotor cortex, there is a higher percentage of cells that lead change in on and other behavior. While in parietal cortex, they are equally distributed, the cells with negative or positive time lags. But I will skip this part, probably not having time. There is time. Yes. Okay, I will not skip it. So, as a control, we also have tested this experiment, making the monkey to interact in a unidirectional fashion. We have basically simulated the joint action task, asking the monkey to perform the task with another, with a fake, with a virtual mate that is represented by the computer, because the computer was moving the cursor in a very similar fashion from the real joint action session. And we have seen that the discrimination power of the cell in the case of the simulated condition is lower with respect to the case of the real condition. And this is true for both premotor and parietal cortex. These are the comparison of the two analyses performed in the real and in the simulation condition. So, since at some point we have noticed from a previous data that from a previous experiment performed on the monkey moving alone, that through a population vector algorithm we applied to the solo cell, we could nicely reconstruct the trajectories of the cursor in a trajectory of the cursor when the monkey was performing the task individually. That is, this is the comparison. The red curves are the neural trajectory reconstructed just from the neural data from something like 175 cells. And these were compared to the gray curves that are the mean trajectory for the monkey. The question was if we could reconstruct, because after all, our main question was to find out a neural representation of the joint action. And for this purpose, we have pooled the activity recorded from the two brains. And we had compared two different subpopulations of cells. The one is, the first was those tuned only during the individual action, and the other tuned during joint action cell. So comparing the two populations, we can see that the performance of the so-called joint action cell, both in premotor and parietal cortex, as it has been evaluated statistically, is much better for this subpopulation of cells compared to the other cells. So the combination of the two separate own kinematics does not provide a good reconstruction of the joint action task, that instead can be better reconstructed by a dedicated set of cells that combine their own kinematics with the constraint that we mentioned at the beginning imposed by the task, particularly by the other agent. Therefore, I will conclude from this data saying that in both premotor and parietal cortex there exists a population of joint action cells that signal the action condition, that is solo versus joint action. In premotor cortex, neural activity of these cells is more predictive than in parietal cortex for both the own and the other action. The population code of solo cells fail to provide an accurate neural representation of joint action. So the neural representation of joint action is not achieved by merging the neural representation of two separate individual kinematics, but rather is achieved to recruitment of a set of cells that integrate signal about their own kinematics and external social cues related to the constraint imposed by the other actor performance. But given the reason we are here, I would like to switch to another take-home message, and to do so, I would like to use the word that are, for me, magnificent and superb word by Vladimir Nabokov, the writer that he wrote at the conclusion of his lectures on literature given at Cornell University. And of course, I will read these few words. The thrill of pure science is just as pleasurable as the pleasure of pure art. The main thing is to experience that tingle in any department of thought or emotion. We are liable to miss the best of life if we do not know how to tingle, if we do not learn to hoist ourselves just a little higher than we generally are in order to sample the rarest and ripest fruit of art which human thought has to offer. And I strongly believe that Nikos knew how to share this tingle among neuroscientists. Thank you.
Okay, it's working. Quick remark on the microphones. Since the techniques don't know which microphone is currently active, they would appreciate if you could just hold up the microphone. It's spreading all over the audience, so it's really hard to tell for them. But if you could just immediately, before you speak, hold it up for 1 or 2 seconds so they know which microphone is to be activated. Thank you. You don't need to.
Well, now we thank you for your wonderful, insightful talk, and I ask for questions.
Yeah, Alexandra, very nice
5
talk.
minutes for questions.
Very nice talk. 2 questions, basically, very quick questions. The monkeys were in the same room, and you compared it with the computer monkey, and then you see that some of the effects go away. If you separate the animals, do you get sort of like similar?
If I separate the two animals,
Yeah.
I'm testing right now. I mean, I did this experiment. This was at the very beginning, and they were working next to each other. And so what I've noticed, and this I also measured, the performance. I noticed that the performance in 3 different couples was much higher when the monkey were working together, instead of when they're working along with the computer. So as if they were happy, and the performance raised up significantly. And now I'm doing the same test, separating the monkey. I didn't see difference so far with a tent, just simply because they can hear the presence of the other. Now I'm just bringing the monkey in another room.
Very, very related question. What if you change the pairs?
Okay, I didn't have I didn't test different pairs because to work with 2 monkeys simultaneously, and I should have 3 monkeys in the lab. I mean, it's not.
No, mingle. No. Just one monkey from the first pair. One monkey from the first pair to the second.
Yes,
pair.
but what I'm saying is that the experiment is done simultaneously, having under-trained 2 monkeys per time, and the other couples are done in another context. What I have done is to simulate this shuffling, to test if the pattern that I noticed were embedded in the task or were related to the fact that I was pairing the right monkey. So I shuffled the trial, and so I simulate these pairs, but not in the real life.
So in
in your introductory slides, you showed us a couple of examples of joint action tasks that cannot be managed by just one agent, like carrying a large sofa. So in the task that you used, that's... Is the microphone not on?
No, it's me, yes.
In the task that you used, it was obviously something that can be realized by just one agent. You not necessarily need the support from the second one. So I was wondering if this might make a difference with respect to the representations you are dealing with. So
Yeah.
What about having a task that really requires that forces need to be added beyond a level that could not be accommodated by just one agent?
Yes, to be honest, because I didn't hear the premises, so I'm not sure to have understood the question, and but it's only a matter of English. So your question is that it's not necessary to have this task.
Yeah, in order to be successful, you just have to take the other one into account, but you not necessarily require a contribution from the other in order to cope with the,
So you mean that it's not necessarily the contribution of the other monkey to succeed in the trial?
in this type of task? Of course, you have to coordinate.
No, I'm sorry to say no, but the idea is that if they do not coordinate, they will not succeed and they will not have their own reward. So, because what I didn't say at the beginning, probably if one monkey goes towards the peripheral target and the other do not move, none of the two monkeys is rewarded. And and this is an error, and this is not a joint action, considered a joint action trial. So in order to succeed, they have to arrive together and in simultaneous fashion, because otherwise the trial is aborted.
I understand, but I would argue it's different from a, well, perhaps we can discuss it.
Okay, I'm very willing to do that.
Okay, let's thank both speakers of this first part of the session and have a coffee break now.